Hello everyone, I am Nilima. Currently I am doing PhD in IISc Bangalore in Molecular Biophysics unit, that is MBU department. Today we will discuss about one technique called silver staining. There are few points about this silver staining, why it is called particularly silver staining. I will discuss about all these points, but normally after gel electrophoresis, that is SDS page, SDS polyacrylamide gel electrophoresis, we do SDS page for the protein separation. Like on the gel we can see the separation of the proteins according to the molecular weight of the protein, and we can detect particularly our protein of interest in gel electrophoresis. So after gel electrophoresis, we generally stain the gel in Comasi Brilliant Blue solution so that we can see the protein band on that gel. But let's say if your protein concentration is not that much, there you can use this silver staining, because this technique is very sensitive in nature and you can detect about 10 to 20 nanogram of protein if it is at all present in the sample. So people generally do silver staining if the protein concentration is very less in the sample. And today we'll discuss about all the steps and chemical which are required in this technique and how this technique works. Actually, this is the most important thing. And after this I will attach one video, that is the practical view how I did the silver staining in the lab. So stay tuned with this. So if we start the discussion of silver staining, the term silver, it came from silver nitrate. Now, in case of Comasi Brilliant Blue staining, we use Comasi Brilliant Blue as a dye, but here we use silver nitrate as the dye. It will go and stain the protein band on the gel. That's why its name is silver staining, as you can see. Now, how it actually works is, let's say you have protein sample. Okay, you have protein sample. Now, if you see on the gel, let's say it's a molecular protein marker, and these are different protein bands. Let's say it's 160 kDa, it's 70 kDa, it's 35 kDa, it's 18 kDa, it's 40 kDa. Like this. These are the molecular marker, and these bands are present. When you will load the protein marker, we can see these bands. These are the molecular marker. Now you have loaded, let's say you have loaded 3 samples. One is, let's say, 75 kDa, it will be somewhere here, and one is, let's say, 10 kDa, it will be somewhere here, and let's say one is 34 kDa, it will be somewhere here. Right? Now, if your protein concentration is high in this sample, you can see these kinds of band after staining with Comasi Brilliant Blue. Okay, but let's say this 10 kDa protein, the concentration of 10 kDa protein is less. Let's say you have 10 nanogram of this protein. So when you load this in this gel SDS page and do the Comasi Brilliant Blue, you can't see the band here. You can't, because the concentration of the protein is very less. So to detect this kind of protein, this lower concentration of protein, we do silver staining. What we generally do in this silver staining, after this SDS page, we generally do not put this gel in that Comasi Brilliant Blue dye solution. Rather, we stain the gel with silver nitrate. Now, how we generally do it and how it works, let's discuss that. Now, silver. So first, I will talk about the mechanism, how it works. Now, silver nitrate is the dye here, and when it reduced, it's form free metallic silver. And what it will do, it will go on the gel, and particularly where your protein is there, it will actually interact with the protein molecule and it will give a dark band. Okay, you can see in my next video, like I told you, I will put one video, the practical view. There you can see the dark band on the gel. Those are actually free metallic silver that interacted with that protein and made these kind of bands. Now, whatever gel I was talking about earlier, let's say this is this is 3 different protein sample. These are the molecular marker, and I was telling that this 10 beta protein that is like in the sample the concentration of this protein is very less. So if we do this silver staining, this free metallic silver will go and bind on that. It will interact with this protein and it will create the band. Now it will also create those other bands as well, because this concentration is very less, and this technique is very sensitive, so we can detect this band as well. Now in this process, in the next video, in the attached video, I also attach that protocol. So in one page I have the protocol, so I put that that protocol as well in the next video. But if we discuss the general steps of this technique, what we generally do, we make different solution. Actually 4 different solution. One is fixing solution, okay, right? One is Na2H2O3 solution, that is sodium thiosulfate solution. One is developing solution, right? And other one is it's called it's citric acid solution. Okay. Now in between these 2 things, I have one that is silver nitrate solution. Okay. Now if we discuss the process step by step, in the first step, what we actually do, we take a box and put the gel here, and we cover the box with aluminum foil, because silver nitrate is actually light sensitive. So whenever you are making the silver nitrate solution, make sure it is covered with aluminum foil, as well as the box is also covered well with the aluminum foil. Okay. So first what we do, we fix the protein on the gel. That's why it's called fixing solution. It's generally contain some glutaraldehyde or formaldehyde, and formaldehyde is the we generally use for the cross linking. So what it generally do, we have formaldehyde or glutaraldehyde, methanol, all these things in the fixing solution. It generally fix this protein. So we can't see the protein in the first step. We can see the protein band after pouring this developing solution. When we develop the gel, that's why it's called developing solution. When we develop the gel, we can see that the band on the gel. But from the first step, we can't see the band on the gel. So first we generally pour this fixing solution in that box. So we have to soak the gel inside this fixing solution, so that your protein it's properly actually fixed on that gel and it stay there. And one tips is here that you can't touch your gel with finger or with hand after pouring this fixing solution, because if you put your finger, your thumb impression will be there on the gel, and I have experienced those kind of faults, those kind of things on the gel. So make sure you don't put your hand or finger on the gel after you pour the fixing solution, otherwise you can see your thumb impression on the gel actually. So then first we fix the gel, and then we remove the fixing solution, okay, and wash it properly with RO water, so that all the fixing solution it's removed properly actually. So after this washing step, you have to put this Na2H2O3 solution here, and after so it's for very very short period, and it's for I think 1 minute. It's that much less time. We need to put this Na2H2O3 solution here, and again, so I'll write it down, Na2H2O3 solution, and then again we have to wash this gel properly. And after this washing step, we put after this washing step, we put this silver nitrate. This silver nitrate solution on the gel and keep it for 10 minutes. and make sure it is covered with aluminum foil properly. So the silver nitrate, it will reduce because it will interact with the protein molecule, and it will reduce and it will form free metallic silver, right? And it will be deposited on that protein sample. And if you see the silver staining gel, no, you can see the band, blackish band, brownish band here and here wherever your protein is there, but other portion will be clean because there is no protein and there is nothing which can actually bind with the silver, free metallic silver. So the other portion of the gel will be clear, but wherever there is protein, it will bind the silver. I just discussed, the silver will go and deposit it on that protein and it will create the band. So now after pouring the silver nitrate solution and after keeping for 10 minutes, we again wash this gel properly, right? So that silver nitrate will be not there. If it's there, when we develop the gel, that time, if you have little bit of silver nitrate there, it will be reduced and it will form the black layer on your gel. So you can't see any band on that gel because your whole gel will look like black, and I have experienced that thing as well. So make sure you clean properly, you wash properly after every step so that the previous molecule is not there at all. After properly washed, when the washing step is done, we will pour this developing solution. Now, developing solution, what it contains actually, it contains this Na2H2O3, it contains the formaldehyde, and it contains one important compound, that is sodium carbonate, that is sodium carbonate. So all these molecules and what quantity you need, that I have mentioned in the next video. So you need the sodium carbonate here, and this developing solution, so what after this, so you pour the silver nitrate, you wash the gel, right? After that you need to pour this developing solution, and so first you have to wash first properly, then you have to pour little bit of this developing solution and decant it, then you have to pour all the developing solution. So first what you did, you wash the gel properly, and you pour little bit, let's say 10 ml of developing solution on the gel and shake it little bit, and then you decant it. After that you pour all the developing solution because it will take little bit time. But if you are seeing your band are coming as well as you pour the developing solution, then you need to put this citric acid solution. So in my video, I have shown you up to this developing solution. After that, you need to put this citric acid solution on that gel. Okay. Now after pouring the developing solution, you just shake it little bit, and you can see the appearance of brownish band on the gel, and that actually is the protein band. It will look like a magic because whenever you will add the developing solution, you can see the band on the gel, and then you have to pour the citric acid solution to finish the process. Because if you over develop your gel will look like black because your protein is there, that is already, like, silver is already is there, that is deposited on the protein, but other than that, other portion of the gel is also will be look like black. So to end that process, you need to add this citric acid solution because here we have sodium carbonate that are salt and basic in nature. Whenever you will add the citric acid there, and you can see there is so it will end the process, and you can see the gel, and you can see the band on the gel properly. So this is the main overview, you can say, or theory portion of the silver staining process. But I have attached one video with this. You can see that video, but that is up to this developing solution. I actually forgot to take the clip of adding the citric acid solution there, but you have to add the citric acid solution after developing the gel, so that it ends the process, and you can see the band, only the band on the gel, not like whole the gel is look like black and something like that. So this is theory portion of the silver staining process, the technique, and you can see the check the other video as well, and all the other video like SDS page and all these things. I have discussed both the theory and the practical, the lab view properly. You can go and check it there as well. Thank you so much. If you like the video, please share and subscribe and share with your friends. That is the more important, because these techniques are something like we generally do in the lab, and you can see other videos like everywhere, but these kind of from the lab direct view, it's generally kind of people don't want to show all these things, but I thought it's might be helpful for you, and if you like it, please like the video or share and subscribe. Thank you so much. Hello everyone, I hope you all are doing well. Today I am back in my YouTube channel with a practical video directly from the lab. So today I am doing silver staining. So it is a staining, it is a SDS page staining procedure. So there are multiple staining because based on your requirement you generally do the staining process. If your protein content is high enough, you can do with Comasi Brilliant Blue, that is fine enough. But if your protein concentration is much lower, then you can do silver staining for your to detect the protein on the SDS gel. So that's why because I'm doing one protein purification, and after purification the protein concentration was very less. So for that you can do silver staining. Now for silver staining we need to make 4 different solutions. One is called fixing solution, one chemical is there, sodium thiosulfate. We need to make a 0.02% solution of that, and we need silver nitrate here, and also we need to make one more solution, that is developing solution, and we need citric acid at the concentration of 2.3 molar. So first what we do, we will make one solution called fixing solution. Now I have already made it. So the composition of this fixing solution is 13.5% formalin, that is formaldehyde, and 40% methanol. So we will make generally make 100 ml or 200 ml based on the gel size, and the composition of the fixing solution is that. Now what I did, I'll show you. This is my gel, this is the SDS Page gel, and I soaked this gel in fixing solution for 10 minutes. Now I will decant this and I will wash it twice for 5 minutes each in normal RO water. Gel as well. So I poured the water and poured the gel as well, and now I am washing. So I kept the bucket, I kept the gel pot in a rock. Okay. So now what we have done till now we did. So we soaked the gel in fixing solution, that is 13.5% formaldehyde and 40% of methanol. I have soaked the gel for 10 minutes. Then I kept for the washing for the first round, that is 5 minutes. So now it is 8:21. We will bring this gel bucket at 8:26, and again I will keep it for the second round of wash for the 5 minutes. So after that we need to make the sodium thiosulfate solution, that is I told you 0.02% of sodium thiosulfate. So that I have already made. You can see the composition here, sodium thiosulfate 0.02%. This is so this normally I make 100 ml for the small gel, but as this is the large gel, so I made 200 ml here. So you can see this protocol. This is the protocol I have taken. You can search on the internet. You can see this protocol. So these are all the composition of those solution, and these are the protocol. So based on this protocol only, I am doing this experiment. So this is the gel. I pour the water for the second round for the washing. So it is for again it is for 5 minutes, and then we will do the next step. Okay, so I have decanted the water which I pour in that box for washing the gel. Now one thing you make sure, after pouring fixing solution, you just make sure you don't touch this gel, because if you just touch this gel, your fingerprint will be on the gel, because this is for fixing the protein in that gel. So make sure you will not touch on the gel after pouring the fixing solution. Now washing is done. Now the next step is I am following the same step which I just already shown to you. So now next step is soak the gel 1 minute in 0.2% Na2H2O3 that solution. Okay, now I have shown you just this is the solution I have made. Now I will pour the solution in the box and keep it for 1 minute. So the time is 8:30. At just 8:31, we will decant this. Okay, during this process, just after pouring that solution, you just shake it properly, shake it like normally, so that the gel is completely dipped into that solution. So 30 minutes remaining. After that, we will decant this and wash this gel for 20 seconds twice. Okay, I'll back after that process. Now I am washing it. So now I am washing it, and during washing also you have to just little bit shake it properly, so that the washing step is done properly, because you need to remove all the Na2H2O3 that solution. You have to remove completely, so that your gel looks clean. Otherwise it will be black, completely black. You can't see any protein. Okay. Now we have already poured Na2H2O3 solution. We have washed the gel. Now the next step is soak the gel 10 minutes in 0.1% AgNO3, that is silver nitrate solution, and that should be make fresh, and the container should properly closed with aluminium foil. That's why we also cover the box with aluminium foil, because silver nitrate is light sensitive compound. So we will make here this is, so I generally make 100 ml, but this is large gel I just told you, so I'll make 200 ml and I'll keep it for 10 minutes in the rocker. Okay, I'll be back after that. So I have poured the silver nitrate here, and now we will keep this box in the rocker for the 10 minutes. Okay, now in these 10 minutes, what we will do, in this 10 minutes we will make the next solution, that is called developing solution, which will develop the gel band on that SDS page. Now for developing solution, we need 3% sodium carbonate, 0.05% formalin, that is formaldehyde, and 0.00016% Na2H2O3 solution. Now what I will do, I will make 100 ml of this solution, that is 3% means 3 gram of sodium carbonate, 0.05% formalin, that is 50 microliter of formalin, and 0.00016% of Na2H2O3, that is, like I calculated that is around 80 microliter. So I will add all these three things in this bottle. This is for developing solution, and I'll make the developing solution. Make sure you are making this solution fresh. It should be fresh, otherwise your experiment will not work. You can't see any band on the gel. So make sure this solution is like just you make and then pour like that, fresh enough. Now 10 minutes is done. So we soaked the gel in silver nitrate solution, that is 0.1% silver nitrate solution, for 10 minutes. Now I will decant the silver nitrate. Make sure you are decanting it in a closed, like this falcon or tube or anything, beaker or conical flask, anything, but it should be covered because it's light sensitive. So you need to discard it properly, not in the sink in your lab, definitely. Now the next step is saying that soak the resin with water, and again with a small volume of developing solution. So I have to first resin this gel with water, and that is RO water, and then I will resin this gel with small amount of developing solution, what I just made. So now first we will do with the water. Okay. So this is with water. Now I will pour this, like decant this water, and now we will pour that is a developing solution, little bit first, and you will see the appearance of protein band on your gel. Okay, just little bit. I will decant this as well, and now I'll pour all the developing solution. Now slowly try to just little bit, you have to shake it. We'll wait for some time. Now you can see the band of the protein.